Okay, so hello everyone. Thank you for coming to this talk. My name is Eli Robles. I'm a nanotechnology engineer, which I'm now doing my PhD here at Oxford at the Department of Physiology, Anatomy, and Genetics. And my research is focused in neuroscience and particularly in Parkinson's disease. So I'll be happy to give you this talk about how both of them intertwine and just how nanotechnology meets neuroscience. So, okay, I really hope. No, okay. Is that better?
It is. Yeah, we just need to hit it again, though.
Yeah.
Perfect.
Okay, perfect. So yeah, this talk is called From Nanobots to Curing Parkinson's Disease. So a little introduction. I'll be talking about how you've probably seen or heard the word of nanotechnology on movies or any other media form, but I'm going to do a comparison of how what we see in the media comes close or similar to what I actually do in my lab every day and how I use that to solve Parkinson's disease treatments and other solutions. So, okay. So this is probably what you encountered when you
sorry. So for some reason, the little warning is still here for us. I thought it would go away after a second, but it hasn't.
The little warning?
Vista del present at all.
Okay. What about now?
It has gone, but the slides are cropped again still. So if you go one slide forwards. Oh, that's better. Perfect.
Is that good?
Yeah, that's good.
Okay. Great. So yeah, and you can see this next slide, right?
Yeah, we can see that. Yeah, the red blood cell.
Okay, perfect. So right, this is probably what you heard or seen when you Google nanotechnology, which seems what you would expect. They're tiny little bots that they could have different applications. So here we see one that kind of looks like a spaceship attacking a bacteria, or another one that looks again like some type of alien machine. But this is not what nanotechnology is, at least yet. Essentially, nanotechnology is a branch of chemistry. So I am a chemist, which works in the brain, but nanotechnology is just a way of using chemistry to make very, very, very small particles that essentially could look like this maybe in the future, but right now they're probably are more like spheres or star shapes. It depends on the application. And I'll be talking more about the type of nanoparticles that I do in my work. So to start off,
stay calm.
Right? To start off, I would like to show you a couple of clips that you've probably seen around different movies, such as Avengers, Spider-Man, James Bond, even on the most recent film, and to kind of compare it to what I actually do. So we're going to start with this Avengers film. Please let me know if you hear it.
I did not enjoy that for a second. Come on, Max.
Where's your gun?
I don't know. Should have been having a thing.
There's no time for a thing. That's the thing right there. Let's go. Dude, you're embarrassing me in front of the wizards.
I'm sorry. I can't or he won't.
It's all right.
Like, I don't.
Hey, stand down. Keep an eye on him. Thank you.
I have it.
Where'd that come from? It's Nanotech. You like it?
Stay calm.
Okay.
Stay calm. Just take a deep breath. Are you okay? No! I promise. I've got this all under.
Oh.
Nanotechnology. You've outdone yourself, Peter.
And the next clip.
What are we looking at?
This is the funeral of one of the dead Spectre agents.
Or contact with the corpse. We found Heracles in the blood samples of all of them. What are they?
They're nanobots. They're nano.
Okay, so I hope you all could hear and see these clips properly. So again, this is what television tell us. They're little tiny machines that could be in our blood cells or in suits, but they're actually not that far from where we are now in research. So let's take a look at it. So I'm going to show you now some real life applications of what nanotechnology actually is. So again, it's not used in Iron Man suits yet, but there are a couple of things that could be applied to, like ferrofluids, that can actually be used for a suit. And this next clip will show you a little bit more about that.
In this vat is a bunch of moving black ooze that's probably the closest thing to venom you've ever seen. Now, if I get close enough, it jumps onto my arm, defying gravity and eventually cover me from head to toe, just like in the movie Venom. So what is this stuff and how is it happening? It's called ferrofluid, and it's got some amazing properties that make it perfect for creating a real-life liquid venom suit. So the reason...
Okay, so this is one of the examples that we can see in nanotechnology. So this is a ferrofluid, which is, it can be nano or not, but if it's nanoparticles, it's more susceptible for it to work better. So why nanotechnology is important here and why can we see it in this specific example? So again, nanoparticles are just teeny tiny particles of matter that essentially most of them look like spheres, but they have different properties. So why is this? So when we enter the nanoscale, matter starts to behave on funny and different ways. So for example, we can have a piece of silver that you just have a block of silver lying in your table. It's not going to do anything for you more than maybe get you a couple of things you can buy with. But if you bring it to a nano size, so around 35 nanometers, very exciting things start to happen. So nanoparticles, silver nanoparticles specifically, have antibacterial and anti-carcinogenic properties. So actually, silver nanoparticles are used every day on kitchens today. So if you go to the supermarket and you get one of those little small bottles that says disinfectant for fruits and vegetables, most of them are actually colloidal silver nanoparticles. And if you extract them and isolate them, you would have yourself a little solution of silver nanoparticles. But again, at this scale, it's mostly used to disinfect different stuff. So also, I don't know if you guys saw when the COVID came out and there were a lot of things with different applications that could help us to get rid of the virus. A lot of them said nanotechnology, and it was because it had silver nanoparticles in it, killing bacteria and other small pathogens. So when we have, again, small particles, in this case, the ferrofluid, it behaves differently. So in this case, we have magnetic nanoparticles. So again, at the quantum scale, matter changes. So the spin of these nanoparticles would change and would enhance some some particle properties that we don't see at a larger scale. So another example of these, it can be used in medicine as well, which I'll show you in the next clip and then explain you a little bit more about the background behind it.
Scientists are using cells isolated from human tumors to develop new ways to tackle cancer. Here we can see the cancer cells growing in a culture dish in the laboratory. For most drugs to be active, they must go inside the cancer cell. Scientists are developing tiny particles, nanoparticles, that can be loaded with drugs. The nanoparticles can be modified so that they attach to the cancer cell and get carried inside. To make sure that the drug-loaded nanoparticles only attack the disease cells, they are engineered so that they only bind to the target cancer cells and not to neighboring healthy cells. Once they bind to the cancer cell, they're taken into the cell. They can't pass through the surface. Instead, part of the cell surface forms a pit, and then it engulfs the nanoparticle. Now we are looking inside the cell. Cellular proteins wrap around the vesicle and drive it inside the cell. You can now see a vesicle carrying the nanoparticle moving towards the very center of the cell. The vesicle carrying the nanoparticle travels further inside the cell and then fuses with a compartment called an endosome. This is part of the digestive system of the cell's stomach. The inside is acidic, and its job is to digest incoming material.
Okay, so the second example is how we can use nanotechnology for drug delivery. So this example is actually most close to what I do in my lab. I don't make Iron Man suits for Parkinson disease, but I do make this type of nanoparticles for neuroscience. So this specific example is used at Monash University, and they use it for cancer to deliver drugs through the nanoparticle. But then again, I use a different type of nanoparticle, and I use it for the brain. So why do we need this type of particles in the brain? Like, why don't we use regular drugs? So in neuroscience, we have this big challenge where a lot of the drugs that we develop in our lab don't always translate into the getting it into the industry so the patient can consume it. And a large part of this, it's because there's this little mesh that covers the brain called the blood-brain barrier. And sometimes getting the drugs through that blood-brain barrier can be very difficult. And here's where nanoparticles come in handy. And again, this all thanks to their size and to their different properties. So the blood-brain barrier has very, very small pores that won't let everything permeate. So this is why we have a hard time trying to get the drugs into it. And again, I have another small video, and this is the last video of the presentation, that will demonstrate how and why we could help to use nanoparticles into the brain and why it's important to tackle it.
Inside the brain, along all of the blood vessels of the circulatory system, there is a highly selective filter called the blood-brain barrier. This barrier allows some substances to pass through into the brain while keeping blood and harmful substances inside the blood vessels. The barrier is made up of several components, the first of which is the tight junctions between endothelial cells, restricting much of what can pass through. The endothelial cells are wrapped in a layer of parasite cells. Those are then embedded in a thick basement membrane made up of a fibrous extracellular matrix. Outside this membrane, astrocyte cells form projections called feet, which further surround the blood vessels. These feet help transport nutrients from the blood to neurons, supporting neural activity. In certain pathologies, like Alzheimer's disease, we believe the barrier begins to break down. This blood-brain barrier dysregulation can occur as endothelial cells degrade, compromising the tight junctions. The parasite cells also degrade, and the blood-brain barrier loses structural integrity. As this happens, the brain's custodial system fails, causing A beta and tau pathology to build up, which are strongly associated with Alzheimer's disease. Over several years, this pathology and the infiltration of other neurotoxic proteins leaking through the broken barrier lead to neurodegeneration, ultimately causing alterations in memory, behavior, and cognition.
Okay, so right, this was an example of what it does in Alzheimer's disease, but then again, I work in Parkinson's disease, and it's quite similar actually to Alzheimer's disease in different areas. But again, they're both neurodegenerative diseases, which I will talk about a little bit more of why Parkinson's disease. So Parkinson's disease is actually the second most frequent neurodegenerative disease, and it affects around 1% of the people worldwide. You've probably seen it around in movies, or maybe you know someone who has Parkinson's disease, and the most recognized symptom is to have a hand that shakes. So again, it's very popular in movies, or nowadays it's getting more frequent. We see it more frequent in patients, and this can be to several factors. Some of them are environmental. So we've seen more epidemiological studies where Parkinson's disease patients come more into the clinic because they've been intoxicated with some heavy metal poisoning or pesticides or other toxins found in well water. So this all can lead to the Parkinson's disease because these toxins do filter the blood-brain barrier, unlike most of the drugs that we're trying to design for them. And they generate chronic stress on the brain and eventually lead to Parkinson's disease. So today, there's not an actual treatment that could stop the progression or cure it, but there are other treatments such as levodopa, which helps for the tremor and other mild symptoms. So this all starts because in 1817, James Parkinson published this essay called The Shaking Palsy. And here's the first time that Parkinson's disease was documented. Of course, we have more years seeing this disease. It doesn't only have 200 years, but this is the first time someone documented it. And it just started to see as a shaking palsy. So again, this tremor that I talked about the hand. And ever since 205 years later today, there's still not a cure. And we haven't really come very close to providing a cure. And again, most of it's because the drugs are not permeating the blood-brain barrier. So what we can see that works in the cells or in some mice models, it might not always work in humans. And this, again, is because the drug is too big for it to permeate the brain. So what I do in my research is I repurpose drugs from other diseases, and I put it in a nanoparticle. So I combine two things to make this nanosystem, which again, I'll talk more in the next slides. So what drug do I use for my nanoparticles? So for the nanoparticles, as a researcher, you have to go through a lot of different scientific papers and some books so we can come up with some novel solutions for the disease that we're tackling. So one day I was reading a paper about liver disease, and I discovered that it was very similar to what I see in Parkinson's disease. So we have something in Parkinson's disease that's a metabolic change, which essentially it's just that the glucose in your cells changes. And this change, again, can be due to an attack of a neurotoxin. So when the neurotoxin, let's say a pesticide, which one of the most common that we see today in Parkinson's disease patients is rotenone. So we can paint a picture where we see a farmer on the rural areas of England that works with the crops, and using this pesticide, he gets infected and he starts to see some symptoms such as the shaking palsy or the moving of the hand. He goes to the clinic and he discovers he has Parkinson's disease. And what happened essentially is that this toxin filtered through his brain and injured the neurons inside of it, as we were seeing on the earlier video, that caused neurodegeneration. So this mechanism, the same mechanism, actually happens similar again in liver disease and even diabetes. So it's very helpful for us researchers to use different types of drugs and other medications that we see that are used in other parts of the bodies, because sometimes, if we're lucky, we can see that it also works in the disease that we're working. So this is what I did. I took this drug that was used in these two diseases and I popped it into some gold nanoparticles. So why gold nanoparticles? So again, as I told you a couple minutes ago, silver nanoparticles are used for antibacterial properties, but gold nanoparticles have a different application. So if I wanted to use silver nanoparticles in my research, it would not be so great because the silver is cytotoxic to the cells. So yes, it's great for killing bacteria, but it's not so great if you want to put it into somebody's brain. because it can be very toxic to it. So I use gold nanoparticles because gold, it's not cytotoxic at all. So it's not going to injure the cells that I mix it up with. So I take my small gold nanoparticles and I combine them with my GLP-1 agonist, which is, well, the medication that I was talking about that I looked from my other papers. And then I just glue them together to make this nano system. So how do I do this? So this is a little video of me in the lab. I wanted to make this experience as like you could be in the lab with me. And making nanoparticles, it's not as hard as it seems. And again, these are just very fancy and long words, but we have to keep in mind that this is chemistry. So we're just making small little parts matter. So it requires a 6-step process, which we mix a salt of the nanoparticle that we're making. So again, in this case, I'm making gold nanoparticles because they're not cytotoxic for the brain. And I do this by getting a salt of gold. So you can order this in, well, what researchers get their regions from. It could be a gold salt, it could be a silver salt, it could be a magnesium salt. But again, we're using gold in this case. So what do we use a salt of the metal? So because it's a salt, it's going to dissociate in water. So what will happen is we're just going to have the other part of the salt in the water, and then we're going to have the gold ions in the water as well. So we're going to have these parts of gold floating in the water. And then what we're doing here is we're going to mix it with an agent that's going to allow us to clump together those atoms of gold into this little spheres, which these little spheres are the nanoparticles. So you just need a salt, the appropriate heat conditions, and another agent to clump up your nanoparticles and to make these little spheres. So when you start seeing the spheres, you're going to notice a change in the color. So again, in this video, we can see it went from clear to blue. And when we see this change of color, it means that our nanoparticles are already forming. And why is it? I mean, gold is not blue, or in other cases, gold nanoparticles can also look red. But what happens is, if we go back to the quantum part, in the quantum world and the small scale, it not only changes properties, but it can also change the way we see light. So this is why the nanoparticles change in color. We have the salt and it looks yellow. And then when we combine it and we clump it up, it changes the solution. It means that our nanoparticles are forming, and we can represent this as a change of light. And this is why the solution changes color. So we just let that react for 2 hours. And then I add another thing extra, which is an amino acid, which is what we can see here. And this amino acid works like a little, we can call it tape, that would help us glue our nanoparticle to the drug. So we can have both of them together. So once we have our nanoparticles glued to our drug, we are going to proceed to put them into some fancy microscopes that we see at our lab every day. So this ranges from different assays that we use to inspect the different part of the nanoparticle to make sure that we have the nanoparticles, the nanoparticle size, and the shape that we need. So this is the most essential test that we use for nanotechnology, size. So if your nanoparticle is not the correct size, it can impact many ways on what you're trying to do with it. So for example, in the brain, if the nanoparticle that I'm using, it's more than 3 nanometers, it might not permeate the blood-brain barrier like I'm expecting to. So I need my nanoparticle to be between 1 and 3 nanometers, which is the smallest nanoparticles that you can make. So to make sure that I have this correct size, and again, because they go into neurons and this will eventually go into people's brain, it's so important that we double check that our nanoparticles are the correct size. We use something called a transmission electron microscope. And this is essentially a big machine that shoots a beam of electrons, and the electrons hit the nanoparticle, and then the beam of electrons that bounce off the nanoparticle, it's what gives us the image of what we're seeing here. and this lets us quantify the size of our nanoparticles. So this is some images I took when I was making the nanoparticles, and this you'll have to do a lot of zooming to see the correct size, but this is one of the most potent microscopes that we have to see nanotechnology. And again, because these nanoparticles are so small, it's a little bit harder to detect than other type of nanoparticles that usually are around 35 to say even 100 nanometers. So going back to the example of the silver nanoparticles that we use for disinfecting our fruit and vegetables every day, those are around 35, 20 to 35 nanometers. So those would be easier to quantify, but because this go into the brain, these are so small, it's a little bit harder to detect, but we do several assays to make sure we're doing the right thing. And this is one of the most important that we use. So what other type of signals we see in nanotechnology? Now, if you haven't seen this type of signals before, that I'm sure many of you haven't, but they're not as hard to see or to learn to detect as other types of microscopy. This actually is quite straightforward. So we use another test called X-ray diffraction, which is again just doing aiming beams at nanoparticles so we can get some type of signal. So let's talk about this one first. So this can look a little scary at first, but then again, it's not. This science, it's full of names with very long names, but don't be scared by it. So what we're seeing here is a signal of our nanoparticle. So because our nanoparticle has this little tape that I talked about, so we can bind it with our drug, it's going to look a little messy. And if it looks messy when we're doing X-ray diffraction, this is good for this type of nanoparticle, because it means that there's an organic solution in our sample. And an organic solution would be an amino acid that we are using as tape to glue both things together. So this is a good signal that we're doing for our nanoparticles. And another thing that we use very common in our lab and that I use every day is the Fourier transform infrared spectroscopy, or just FTRA. And then again, don't be intimidated by this name. These are just long names. And if you look at it, it's just a line. And if you learn to read these lines, you're going to be a great nanotechnology engineer someday. What we're seeing here is another laser being shot on nanoparticles, essentially. And this is easy to interpret if you have a database. So if you become a researcher and you go down this line, which I really hope you do, you're going to see a lot of patterns. So this is like the fingerprint of our metal, or like the fingerprint of our organic material. So you'll learn to quantify how many, it's most about how many peaks you see in here. And depending on the number of peaks, it's going to tell you what material you're most likely are having. So let's say this has 20 peaks, then we're probably going to have, if we look into the library, a combination of amino acid with our gold nanoparticle. Again, this is just more FTR, so you can see the difference. So this is a zoom in of what we had here and added with this other signal. So this LNIPC is our amino acid, which is again our tape we can glue our drug to. And when we combine it with gold, you see that this little peak decreases and we have a flat line. If we have a flat line like we have here, it means that our nanoparticle is binded to our amino acid and the solution was successful. So, okay, now that we have our nanoparticles in place, it's time to do some cell treatment. So what you want to do after you confirm your nanomaterial, you want to go ahead and treat your cells with it and make sure your experiments are aligned with the disease you're trying to model. So in this case, again, I'm working with Parkinson's disease. So I'll use 4 different types of parts of the experiment for this. So what you always want to have as not only as a nanotechnology engineer, but also as a researcher, you always want to have a control group, which is the baseline of your study. So this is very important in science because this will tell you the comparison to see if your treatment works. Because you need something, it's like a blank. You need to see if something changes. So this is a very important to have. And if you're ever going to a lab and you're doing science, you always have to have a control. And then we have 6-HTA. So 6-HTA stands for 6-hydrodopamine, and this is the toxin that we use to model Parkinson's disease. So I'll talk a little bit more in the next slides, but it's not so commonly known that you can grow neurons on a little dish in a lab. So it's not just what you have in your brain, but essentially you could take some neurons out of this. Typically comes from a patient, or it could come from post-mortem studies, or it could come from a donor, or it could come from a rat. So it depends a lot of where we can get our cells. But let's suppose this comes from a patient, maybe a patient that recently died out of any disease, and the tissue was fresh enough that we can isolate it. And how scientists isolate cells is you take a little piece of the brain by surgery, and then you use some chemicals to digest the cells so you can have this solution of cells, because essentially tissue, it's just a combination of cells, right? So if you digested them, you can have single cells within your solution. So you take a piece of the brain, you digest it on a little tube, and you have your solution of cells, which I'm going to show you how it looks like in here. So this is how the cells looks. These are cells that have been isolated from tissue, from patient's tissue. And we keep them on this big bottle over here that's full of liquid nitrogen. So we can keep the cells, they're not, we'll make them live when we put them into some cell media solution. But essentially, these are just freeze. And again, we go back to the movies because science can be really cool and can look a lot like movies. So when I don't know if you've probably seen this futuristic movie where they freeze somebody's bodies down so they can live, I don't know, like 100 years later. They just unfreeze their body and it's like nothing happened and the person is live again. Well, this happens with cells at our lab. So you take the cells of the patient, you freeze them down at a super cold solution, which I think this is minus 80 Celsius, and then you freeze them with liquid nitrogen, and then you just take them out and you can use them again. So when we take them out, we put them in this little cube tray so we can unfreeze them. We thaw them by using a warm solution so the cells are happy because, again, they just been stashed into this huge cold solution for months, and we keep them, some of them even for years. So you want to keep your cells happy, and essentially cells can become kind of like a pet because you have to keep feeding them and take care of them so they make sure they grow properly into neurons. So once we have the solution and we take them out, we put them onto a little dish and we give them what it's like a mix of nutrients so we can grow them properly and the tissue starts to regenerate. So this is how they look. We take them and we put them on a plate, and this was taken under a microscope, and we stain them with, we can use different dyes depending on the experiment, but this is one of the most popular dyes used in biology, and it's called DAPI. And DAPI stands for the nuclei of live cells. So all of those blue dots that you're seeing are the cells that I grew that I just talked about here. We just take them out, put them on a plate, add some paint into it, essentially, and pop them onto a microscope. And this is how they look. This is how neurons look, which is so exciting to know. You can hold a culture of neurons in your hand and see them under the microscope. It's incredible. So this is how the cell looks. This is how our control line looks, which is just regular cells, nothing on them, like I just grew them in the plate. And this is what happens to the cells when you add the toxin on it. So again, by adding 6-HDA, we're causing Parkinson's disease to the cells. So these neurons have Parkinson's disease, or a model of Parkinson's disease, as close as we can cause Parkinson's disease to a group of neurons on a plate. And when you add this toxin, we can see most of them die. So this toxin is just very harmful for this specific type of neurons. So they die. And when we add, so N is our nanoparticle and E is our drug. So when you add the drug to the cells, you can see that they're not cytotoxic, which is what I was explaining earlier. So gold is not going to do anything harmful to your cells. It's just going to float around. It's not going to react with everything. It's just going to float around the brain, which is very good when we want to transport drugs across the blood-brain barrier. So we can see that the drug is not toxic to our cells. They look pretty happy. They look pretty similar to our control line. And when we add the toxin plus the drug treatment, they look like this. So all of these group of cells are the neurons that have been treated for 3 days straight. So 3 days, for 3 days straight, I went into the lab. I added 6-HDA to this. I didn't add anything to this ones. And for this, I just added the drug. And for this last group, I added the toxin and the drug. So what happens here? Well, these all look quite similar. So we can see that's not only non-cytotoxic, it actually brings the neurons back. It aims for neuroprotection, so the cells don't die. And under some other experiments that were performed, we can also see that it regenerates the neurons. And why are we seeing this? Why? So again, because our nanoparticles are so small, we're working this so we can do it in humans and put it across the blood-brain barrier. But here we just have a plate of neurons. But even though if we just added some very big nanoparticles, or well, as big as nanoparticles can be, let's say 100 nanometer nanoparticles, and I just put them on a plate, they're not going to do anything. They're just going to float around and not really get into the neuron because the nanoparticle is not going to be small enough. So here, the size of the nanoparticle is so dependent on the results we're getting, because again, these are 1 to 3 nanometer nanoparticles. So they're not only small enough that they permeate the blood-brain barrier, that's small enough so they can actually permeate the cell as well, which is what we were seeing on one of the videos earlier in the presentation on the Monash University that they were using for cancer. You can see that those nanoparticles were getting into the center of the cell. And again, this is all depending to the size. So chemistry essentially can help us do this. So by adding the nanoparticle with the drug, we can get them into the neurons, protect them, and even retrieve or promote neurogenesis, so we can actually keep growing neurons. So this is very exciting as a solution for Parkinson's disease, because we're not only aiming to cure aiming to treat the symptoms that I was talking about earlier, which is the shaking palsy or just the shaking of the hand or any other mild symptoms that the Parkinson's disease patient can have. We're aiming to cure it here. We can actually provide something that would be good enough for the cell, that can actually get into the cell properly, that can find a cure for Parkinson's disease. So yeah, this is my research, and this is what I work every day. So right, in this part of the slide, I'm just going to tell you the conclusions. So again, we attach the amino acid into the small tiny nanoparticles that would allow us to glue the GLP-1 agonist, or well, the drug, into the nanosystem. And we also measured it with different types of assays, such as Neuroland, DAPI, which is this what I was showing you with. And the nanosystem kept cells alive by neuroprotection and promoted neurogenesis, so we can keep growing our tissue. So again, this is exciting, not because it can only protect the neurons and stop the progression, but it can also potentially reverse it. And this was all done in the Laboratory of Molecular Neurodegeneration here at Oxford, at the Kavli Institute, and at my lab, the Oxford Parkinson Disease Center. And oh, I had one more slide, but it's not showing. But okay, so I guess I'll just tell you. But I had one final slide telling you that this is how I use nanoparticles to, well, be a hero in the medical sector. But you can use nanoparticles, as we talked about earlier, in the ferrofluid and make maybe the next spacesuit, maybe not the Iron Man suit yet, or maybe in 100 years, someone is going to come out with an Iron Man suit. But you can make the next spacesuits. You could go into Tesla. You could go into many other different exciting technology industries and break the knowledge barrier. Just go beyond it. You can be a hero by making spacesuits, or you can be a hero in medicine and make exciting treatments that would help patients all over the world with different and challenging diseases that we see more and more every day in our society. And again, Parkinson's disease has augmented in case drastically since we came across climate change, because many of these new fancy diseases are because of toxins that we find in air or in well water. So I hope this talk inspires you to maybe take a deep dive into nanotechnology literature, because again, it's not so far from what we see in the movies that maybe seems crazy and it seems fancy and it seems fictional, but we're not so far from it. And going into this line of research, as fancy as it may sound, it's very fun, and anyone can do it. Anyone who has the motivation and the exciting to go beyond and make a change, everyone can do it. And it's a very exciting and rewarding area to be in, to see and to change a little part of the world. And I would like to close by one of my favorite quotes by Gandhi, which is, "Be the change you want to see in the world." Thank you. This is the end of my slides, but I'd be happy to take any questions about nanotechnology and well, neuroscience. Okay, I see some questions on the chat. I think I'm going to start with those. Okay, so Sam asks, do you have the paper I could read on this? Also, when the human trials happen, when might a treatment come online? Oh, okay, so this is a very nice question. Yes, I have all these papers. So the papers that I used, well, I took screenshots of these papers on the presentation, but I can send you, I think I can put it all together in an email of the papers that I used for my research and other exciting reviews. Reviews are so good to use for learning about a new subject. So if some of you don't know where you can find reviews, it's as easy as going into Google Scholar and just typing up nanotechnology or nanotechnology and neuroscience, and you'll come up with this big, maybe 40 pages scientific documents that will give you essentially a review of what's been doing today for nanotechnology and neuroscience. But yeah, I'll come up with the list of papers that I've been using, and I'll make sure you can get that into the email after the list. And the other part of the question was, when will human trials happen and when might a treatment come online? So at least for my part, right now I'm performing more experiments on cell lines, but I'm hoping that by the end of this year, probably on August-ish, well, middle of the year, I'm planning on some mice models. So going into human trials is not the easiest thing. And again, we can do a lot of things on cells and we can do some things on mice, but going into human, it's a new challenge because we're dealing with people's lives here. So this is serious. And before we get to the experiments in humans, we have to have cell experiments and mice models or animal model experiments. So I have the cells experiments, check. Now, at the end of the year, I'll be working with some mice models so I can compile my data and submit that for, well, starting to approval for do human trials, but not yet. Again, hopefully I have my experiments at the end of the year, mice experiments, and then when I'm done with those data, I will submit it into a clinical trial. But we're talking about it's difficult to say, but essentially it takes about 5 to 10 years to get a drug that you're working into the market. Now, because nanoparticles have been approved before by the FDA and other regulatory organizations, I'm hoping it come more smoothly. And also GLP-1 agonist, the drug that I'm using for the nanoparticle, has also been approved in other diseases. It's actually being used in diabetes every day. So we know that these are safe for humans, both the nanoparticles, gold nanoparticles, and the GLP-1 agonist. So hopefully, I mean, we've seen in cells that they don't cause any damage. Like on the contrary, they generate neurogenesis. So we hope that the transition goes more smoothly because we're using two things that have already shown to be good to humans before. But again, we're talking at least 5 years. So fingers crossed. Okay, next question. This is by Adele Stewart. Besides sizes and nanoparticles and permeability, what other issues have you encountered when attempting to find a treatment for Parkinson's disease? Oh, this is actually a very good question. So a lot of the drugs that are being used in clinical trials at the moment, not mine, but other drugs at other labs, don't really tackle what I think is the center of Parkinson's disease. So I'm using a GLP-1 agonist because I believe that Parkinson's disease starts at the mitochondria. And what the GLP-1 agonist does, it aids the mitochondria to help cellular respiration. So I'm doing more of a metabolic effect. So if you haven't heard these terms before, again, just fancy science terms, but essentially mitochondria is the organelle of the cell that produces ATP, which is our energy source as humans. So when the toxin comes, it has been shown that it always, or in most cases, it attacks the mitochondria. So because the mitochondria is producing our energy, the body goes crazy and goes into a process to try to keep the homeostasis and the cell levels at what they should be. And it starts recruiting other proteins or other enzymes, and it causes a huge disbalance. And I believe we can cure Parkinson's disease by aiding that disbalance in the mitochondria by using a GLP-1 agonist, because a GLP-1 agonist helps regulate metabolic function. So we can tackle it at the root. I think we can be pretty sure we can actually cure Parkinson's disease. And again, there are different approaches. The thing is, there are a lot of gaps in the Parkinson's disease research. So not everyone knows, well, no one knows why Parkinson's disease starts. Like we have a lot of evidence and we have a lot of data, but there's not like this whole map of how it exactly starts. Not like cancer. There's a lot of research in cancer, and yet again, we have a lot of questions about it. Even more so in Parkinson's disease and neurodegeneration. We don't know exactly how it starts. There are different theories out there. Everyone goes by their gut, by their scientific gut, I should say, by their educated guess. And my educated guess is that starts with the mitochondria. So this is why I'm using this approach. But there are other scientists that believe different than me, and they have different results than what I have. So there a couple of colleagues that I have working here that are using lipids for Parkinson's disease, that again, they're repurposing a drug that is used in cancer, and they hope it can work on Parkinson's, but they're not seeing such as good results, honestly, as they could. It's not, it's stopping the progression, but it's not generating more neurons. It's not retrieving the disease. So pros and cons. Again, different approaches, different minds, because we don't have the answer right now. Everyone's going by their educated guess. So yeah, it's the challenge of the gap information, I guess. Okay, next question. Sami Jen, how exactly does the amino acid glue the nanoparticle and the drug together? Okay, this is also a very good question. So the amino acid has a thiol group, which is just a chemical group with sulfur and hydrogen. So this thiol group binds to another part of the peptide. So my drug, the GLP-1 agonist, it's a peptide. So the thiol group binds to the peptide, and they make like this type of Lego pieces, if you like. They're just two blocks that bind together chemically, because the atoms are in the right place, the electrons, and it's a lot. We would have to jump a lot into more atomic sense of the talk, which I'd be happy to do. If you want more resources about it, I'd be happy to send it in an email, or just send me an email, and I would like to talk about it more through following that conversation. But this is a very good question. So essentially, these two chemical pieces bond together, and amino acids with thiol groups are very used in drug delivery, because the thiol groups, they just bind to peptides. Again, they have these two pieces that connect, they work. So I'm using L and IBC, but there are other amino acids that can be used. But as long as it has a thiol group and you're using a peptide, you're probably good to go. Of course, you should also do some research first before you start combining these things, but mostly they work like that. Okay, next question, Georgina. My mom has platinum coils in her brain, and I wonder why we use gold rather than platinum. Oh, so actually, platinum is actually a very interesting metal as well, and it's used widely in medicine too, in different applications. But I mean, it's great because it's not cytotoxic as well. Unlike silver, again, silver, we said it's cytotoxic, so you don't want to have that in the brain. At least not at nanoparticle scale. Because then again, things change, funny things happen in the quantum world. So again, I imagine these platinum coils that your mom has are not nano-sized, so they're probably going to have different properties than nanoparticles do. So I know that platinum nanoparticles have been used in a range of different applications, such as catalyst. So I've seen some research where they have like this little plate with platinum on it, and also they usually add another metal. So I think titanium is also very widely used in catalysis features. So they use this to make biofuel or any other catalytic reaction. But sorry, I went too far from your question. But why do I use gold and not non-platinum? So actually, not just because platinum nanoparticles have different properties that magnum platinum, but also because gold is just cheaper to get that platinum. Platinum is so expensive if you want to use it in the lab. But gold is actually more easier to get, and it's not cytotoxic. And platinum, it's not only expensive, but at a nanoparticle scale, because they have this catalytic features, they might be more cytotoxic than other type of metals, and they might kill neurons in this case. Yeah. Okay, we have another question.
Ellie, sorry, can I jump in, because we're getting quite close to the end here, and there's a lot of questions left. I don't want to get through them all, unfortunately. But I have noticed two that I think would be really nice to end on. There's one that asks about your favourite aspects of working in research, and what do you not like about working in research. If you answer that one, and then I'll jump in with the last one.
Okay, sure. Thank you, Jack. So my favorite aspect about working in research, honestly, everything. I love coming into the lab every day. I love that this is my work. when I first, I'll go quickly because I know we don't have a lot of time, but essentially I was going to go into business school because my parents wanted me to, and then I came across chemistry and felt like play to me. I love it so much. And I thought, I don't know, I thought, oh, this is a cool hobby to have. And then when I figured out I can actually make a living out of it and help people with it, just sold me because this is so exciting. I mean, having neurons on a dish and see it every day on your lab, making nanoparticles and see how they change. I mean, this is all very crazy, cool stuff that you see in movies, but you get to see this um, every day on your work. So it's very, very exciting. Uh, Everything it's like magic. Honestly, I love coming to the lab. I live in the lab. It's an amazing place. And being an academic, you get a lot of freedom to when you want to work. So that's also, as long as you have to get your work done, of course, but you have a lot of freedom with that. So I love it. And something I don't like, maybe it can be slightly stressful from time to time, but honestly, if you love it, you don't see it as much. Just regulate your time properly and you'll be good to go. And right, and the other question, sorry, Jack, which was it?
The other question was, it was about undergraduate degrees that would lead into a career in nanotechnology, but I think we'll expand on that a little bit. So is there anything you would say is helpful if you want to go into this career? Undergraduate degrees, but also like what else would you suggest to do if you find this interesting and want to make it into a career going forwards?
Okay, so quickly. So actually my bachelor's is an undergrad. It was nanotechnology engineering. So I'm from Mexico. So in Mexico, you could do nanotechnology engineering as a bachelor's. I know it's not so common in the United States and other parts of the world, but for me, my whole bachelor's was nanotechnology engineering. And if you have a different career right now, or you don't have that option to get a nanotechnology engineer bachelor's, material science is very close to nanotechnology engineering. Chemical engineering, it's not so much. Actually, it's kind of contradictory, but it's more towards actual like pipes and stuff. But probably material science or any biotechnology based if you want to go into medicine, and electrical engineering if you want to go into more like the ferrofluids or Ironman kind of style. Yes, take courses and go to congresses a lot. You'll get a lot of resources from that. Yeah.
That's brilliant, and we're exactly on time. We unfortunately can't answer all the questions now. There was loads and loads of questions, which just sort of, stands testament to how interesting and how well presented this was. I have mentioned this in the chat, but if you do have more questions or we didn't have a chance to answer your question today, if you email outreach@balliol.ox.acu.uk, we will pass them along to Ellie, and if Ellie's kind enough and happy enough to do this, then she'll answer them, and then we'll send you all like a long list of questions and their answers in the follow-up email, because I think there is a lot of interest here that we don't want to leave you hanging, so we will come back to you. But yeah, I mean, that is it from us. We have to stick to time, unfortunately, because these are being recorded. But yeah, thank you so much, Ellie, and thank you everybody for listening and for the quality of your questions. They're really interesting to listen to. But yeah, good night, everyone, and thank you very much.
Thank you. Thank you for coming.